Hey guys, Miss Cote here, and we're continuing the biochemistry unit with this extremely exciting episode about lipids. So let's get to it. The second class of biological molecules are the lipids. Remember, these are not considered true polymers, and that's because they are not made of just one type of monomer like the other three. Lipids are pretty diverse, but the one thing they have in common is that they are hydrophobic. They do not like water, hence the cartoon on the previous slide. Like carbohydrates, lipids are made up of carbon, hydrogen, and oxygen. However, they don't have as many oxygen atoms as carbohydrates. So a molecule of mostly carbon and hydrogen are known as hydrocarbons, which form nonpolar covalent bonds, which makes it hydrophobic. See, we'll constantly be making connections back to chemistry, which is why I have you do that unit as a summer assignment. There are four main classes of lipids. Your book talks about fats, phospholipids, and steroids. But also remember that waxes are also considered to be lipids. So let's start by discussing fats. Fats are composed of two types of molecules, glycerol and fatty acids. Glycerol is a 3-carbon molecule with a hydroxyl group, remember your functional groups, attached to each carbon. This gives glycerol the ability to bind to 3 other molecules. In the case of fats, molecules called fatty acids take up the 3 binding sites. Fatty acids, shown here in the yellow shaded area, are long hydrocarbon chains with a carboxyl group at one end, the COOH group. The OH, hydroxyl group at the carboxyl end, is what will join with the hydrogen from the hydroxyl group of glycerol to make water, which is released in this dehydration reaction. When the 3 binding sites of glycerol are joined to 3 fatty acids, we get this molecule that we see here. The covalent bonds that are made are called ester linkages, another bond name you'll need to know for this class. The final fat molecule, composed of glycerol and 3 fatty acids, can be called a triacylglycerol or triglyceride. Notice that the 3 fatty acids do not need to be the same. Don't get me wrong, they can be, but it's not required. And while we're on the subject of fatty acids, while they can differ in the number of carbons in the skeleton, they can also differ in the number and locations of double bonds. You'll need to understand the differences between the two and be able to recognize which is which. Saturated fatty acids have no double bonds between the carbons. Saturated means they have the maximum number of hydrogens that are allowed. Remember that carbon can form 4 covalent bonds with other atoms. Just so you know, this is the shortcut way to represent the carbon chain. It's understood that there is a carbon every time the line changes direction, and the maximum number of carbons are also assumed. You will see when we do our activity that writing out all the carbons and hydrogens creates a jumbled mess. When looking at a space-filling model of saturated fatty acids, it forms a straight line. These fatty acids tend to be solid at room temperature, and most animal fats are saturated. Think butter or the white squishy fat that you cut off a raw piece of meat before or after you cook it. Unsaturated fatty acids have at least one double bond between the carbons and therefore has less hydrogen atoms. One double bond is called a monosaturated fatty acid, while more than one double bond is called a polyunsaturated fatty acid. Having a double bond in the structure causes a kink or bend in the hydrocarbon chain as shown in the space-filling model. These fatty acids tend to be liquid at room temperature and are generally plant or fish fats. There are certain unsaturated fatty acids that our body cannot make, so we need to obtain them from our diet. One example are the omega-3 fatty acids that you get from eating fish. These fatty acids are required for normal growth and can also protect you from cardiovascular disease. And if you don't like fish, you can always take omega-3 supplements. The function of fats is for long-term energy storage. We can store more energy in a carbon-hydrogen bond than in a carbon-oxygen bond, so fats are a more compact way to carry around extra stored energy. Basically, it takes up less room than storing the same amount of energy in glycogen. Extra fat is stored in specialized cells called adipose cells, which can swell or shrink as fat is deposited or taken out. In addition to storing energy, having fats in adipose cells can create a cushion to protect organs and provide insulation, which is good if you're a mammal that lives in cold water, like a whale. The second class of lipids are the phospholipids. The structure of a phospholipid is a glycerol attached to two fatty acids and a phosphate group taking up that third binding site. And while the two fatty acids are hydrophobic, the phosphate group gives that end of the molecule polar or hydrophilic tendencies. Hopefully you remember why phospholipids are extremely important to cells, because they make up the cell membrane. You know, that whole phospholipid bilayer thing. Let's look at a couple of models. Hopefully, this model looks familiar to you. The hydrophilic head is represented by the circle, and the hydrophobic tails are the little legs. When you add these phospholipids to water, they will arrange themselves into two layers, a bilayer, so that the hydrophilic heads, the part that likes water, will face the water, and the hydrophobic tails, the part that doesn't like the water, will face away from the water. The phospholipid bilayer forms the boundary between the cell and its external environment. We'll discuss the cell membrane in more detail when we get to cell transport. And looking closer at a phospholipid, you can see that two sites on the glycerol molecule are covalently bonded to fatty acids. Here, one is saturated and one is unsaturated. This part of the phospholipid is hydrophobic. The third site is bound to the phosphate choline complex, which have charges on it, making this portion of the phospholipid hydrophilic. Steroids are the third class of lipids. Steroids have a different structure from what we've been looking at, but are easily recognizable with the four fused rings that have various chemical groups coming off of them. The type and location of the chemical groups is what makes each steroid different from each other. All steroids are derived from cholesterol, and while our liver can make cholesterol, we also get cholesterol as part of our diet. Cholesterol is also a component of the cell membrane as well. However, too much cholesterol can lead to cardiovascular disease. And lastly, the fourth class of lipids is the waxes, which your book doesn't talk about, but I'd like to mention them because I think they're pretty important. Waxes are made up of a long-chain alcohol and a fatty acid. As with all lipids, they are water insoluble. Think about how water beads up and rolls off of a freshly waxed car. And if you think about the wax you may rub on a car after you wash it, it's generally solid at room temperature. In biology, waxes are mainly used for waterproofing. Many plants have leaves and fruits with wax coatings to not only help prevent water loss, but to also prevent too much water from getting in. Think about the delicate tissues of leaves. If they're exposed to a lot of rain, water would make the leaves soggy and they would probably fall apart. Some animals also have wax-coated fur or feathers to repel water. A bird who has water-soaked feathers on their wings would not be able to fly, and that wouldn't necessarily be a good thing. And that's about it as far as lipids are concerned. The next episode in this series will be proteins. Again, if you have any questions, please feel free to bring them up in class. So until next time, let's be good humans.